Mapping and sequencing genomes. This main topic, it is divided into two parts. पहले हिस्से में हम देखेंगे कि genome mapping क्या होती है, and in second portion we will discuss that what is genome sequencing. Although sequencing we have discussed previously, but those are some basic techniques, and here we will elaborate it in a few further topics. But first we are going to start with the mapping of the genomes. So the first topic is genome mapping. Genome mapping is the process by which a specific gene it is being designated or assigned to a particular region of the chromosome. And then after assigning particular location, then we can also point out that what are the relative distances between different genes that are located in the genome or on a chromosome. जीनोम मैपिंग एक ऐसी टेक्निक है जिससे ना सिर्फ हम जीन्स की लोकेशन को पिन पॉइंट कर सकते हैं क्रोमोसोम्स के ऊपर, बल्कि उनके दरमियान जो रिलेटिव डिस्टेंसेस हैं या डिस्टेंसेस इन टर्म्स ऑफ न्यूक्लियोटाइड बेस पेयर्स वो भी हम डिटरमिन कर सकते हैं. जीनोम मैपिंग की जो है वो दो मेजर टाइप्स हैं, लाइक द लिंकेज मैपिंग और द फिजिकल मैपिंग. जो लिंकेज मैपिंग है इसमें हम डेमोन्स्ट्रेट करते हैं कि जीन्स का जो ऑर्डर है वो क्या है और उनके दरमियान जो रिलेटिव डिस्टेंसेस हैं वो कितने हैं. एंड ऑन द अदर हैंड, इन केस ऑफ फिजिकल मैपिंग, we can exactly pinpoint the locations of the gene, that this is the particular point where the gene is located, and the distances among different genes can also be calculated in terms of number of nucleotide bases. So in case of linkage map, it just only gives the relative positions of the genes and relative distances among them, but in case of physical mapping, here we can exactly pinpoint the location of the gene and calculate the distance among them in terms of nucleotide base pairs. We can say that genome mapping allows us to navigate around the genome. If we compare such mapping of genome with Earth, then we can understand it in a better way. जैसे हम अर्थ का के मैप्स का एनालिसिस करें तो हमें हमारे नॉलेज में आएगा कि द मैप्स दे मे हैव कंस्ट्रक्टेड ऑन द बेसिस ऑफ डिफरेंट क्राइटेरिया, कि कभी लेंथ यूज़ की गई है, विड्थ, लॉन्गिट्यूड और लैटिट्यूड्स. सो मैप हैव बीन कंस्ट्रक्टेड एंड डिफरेंट लैंडमार्क्स कैन बी असाइंड ऑन द अर्थ, एंड दोज लैंडमार्क्स दे कैन हेल्प टू फाइंड डिफरेंट लोकेशंस ऑन द अर्थ. फॉर एग्ज़ाम्पल, अगर हम अपने कंट्री की पाकिस्तान की मिसाल दें तो इसके जो सदर्न पार्ट्स हैं उसमें ओशन है, देन फर्दर टुवर्ड्स द नॉर्थ मे बी डेजर्ट इज़ देयर लाइक द थार डेजर्ट, एंड देन सेंट्रल पाकिस्तान हैविंग मोर और लेस प्लेन एरिया, एंड देन वी कैन मूव ऑन टू पोटोहार हैविंग सेमी एरिड climatic conditions, and then northern area it is characterized by having lush green forest and valleys. So these are the different landmarks in the country, and on the basis of these landmarks we can exactly pinpoint the locations of certain territories. So in this way we can move anywhere within the country, and same at the global level. So different maps have been constructed, and different landmarks on the earth, they can help us to navigate around. So similarly, such landmarks, they are also existed on the genome. Now one can ask a question, what type of landmarks on the genome? So here, landmark may be any part of the DNA. मिसाल के तौर पर अगर हमें किसी डीएनए सीक्वेंस का जो है लोकेशन का पता है, उसके सीक्वेंस का पता है, सो सच डीएनए सीक्वेंसेस They can act as a landmark or genetic markers, some regulatory sites that can turn the expression of some genes on or off. They can also be the landmarks, even genes themselves. In addition to that, non-coding region, they can also act as landmarks on the genome. So, if the location of a particular landmark or the genetic marker on the genome is known, then the position of other genes in relation to that known location, it can be characterized. हमें पता है कि जो अर्थ का मैप है वो इस तरह का होगा, इस क्राइटेरिया पे बेस करेगा. अगर हम इसको कंपेयर करें जीनोम मैप्स के साथ, तो जीनोम मैप जो होता है ये मोर और लेस वन डायमेंशनल होता है, लाइक द स्ट्रिंग ऑफ द डीएनए, लीनियर स्ट्रक्चर ऑफ द डीएनए. इसको अगर हम एक रोड की फॉर्म में शो करें मैप के ऊपर, जैसे एक हाईवे मूव करती है तो रास्ते में आफ्टर इरेगुलर इंटरवल्स डिफरेंट सिटीज या टाउन आते हैं, तो इन द सेम वे द जीनोम हैविंग द मैप दैट इज वन डायमेंशनल. So different genes, they are located at irregular intervals, and they may be given different naming. Now we can also point out that what is the difference between genome map or genome sequence. We can say that these two activities, although are portraits, belongs to the genome, but they may be separated. For example, if we have constructed the genome map, it will not tell us about the sequencing. It will not going to give any information about the sequence or the nucleotide bases on the DNA. And similarly, genome sequence, it will not give any information more or less about the genome mapping. So both these two activities, they may be separated from each other, but even then there are some close relationship, like two neighbors, they are living together. And sometime what happens that a known sequence of the DNA segment, it can act as a landmark on the genome. So one can say that कि जो genome sequence होता है ये एक detailed genome map होता है. So in this way they may be very closely related to each other. Another additional point is that if we know the sequence of the genome, then what is the need of mapping? मिसाल के तौर पर आजकल बहुत सारे ऑर्गेनिजम्स का जो जीनोम है उसकी जो सीक्वेंस है वो कंप्लीट हो चुका है, इंक्लूडिंग आवर ह्यूमन जीनोम, genome of some model plants like Arabidopsis, some prokaryotic microbes. Most famous one it is the E. coli. So genome sequence is known, full length sequence is available. Then what is the need of genome mapping? Because we have the sequencing already in place. So under such situations, one can say that genome sequencing, it helps, or the genome map, it helps to sequence the genome. अगर हमें genome mapping का पता है तो different landmarks जो हैं इसकी sequencing में help करेंगे. In addition to that, if sequence is available, then it will just be a featureless. Featureless means that we don't know where a particular gene is located, whether this portion is coding or junk region of the chromosome or genome. So for this purpose, we have to designate certain locations with reference to the position of certain genes. So because of this reason, although sequencing is available, but we need to mark some areas on the chromosome. So finally, we can say that genome map, it pinpoints the certain features on the organism genome, and it is very, very valuable tool to study certain genetic diseases, morphological traits, and comparing of different genomes, and of course it is also going to help in the genome sequencing.